Cool. Okay, so we'll go over questions 12 and 13. Let me share my screen. Can you see that all right?
Yes.
Okay, awesome. I just wanted to double check because I'm doing like a split screen type thing because I usually don't take my office hours from home.
Oh,
So
no, I mean, I see the canvas screen.
perfect. Okay, so 12 and 13. Okay, so to start, all right, so you're given the mRNA sequence, which is,
So,
uh-huh,
so we at the office hours with Dr. Holchik today, we did the activity one, where it kind of goes over the mRNA and the tRNA and then the amino acids, the three letter, one letter, all that. So
yes.
Dr. Holchik did do like the first two of each section. So from
Okay.
what I understand is the mRNA sequence that's given. Um, so I just start there, right? And I turn it into tRNA, right? Before I do the amino acids.
No, so you'll be using this mRNA sequence directly.
Oh. How so? Why is that?
Oh, why is that? So this
Yeah.
codon table, um,
Yeah.
these sequences are also 5 prime to 3 prime. So if you were to use the tRNA anticodon sequence, that would be 3
Yeah.
3 prime to 5 prime. Makes sense. I don't
Oh.
know why they like decided codon tables should be mRNA sequences, maybe since it's just easier to get mRNA first, but you'll be using mRNA. So like as you know, methionine is the start codon all the time. So here you
Mm,
have AUG as the mRNA
yeah.
sequence, like the first codon, and that's going to be for methionine.
Oh.
Yeah.
Okay. So the table that's here, is that always 5 to 3?
Yes. Yes.
Okay. Okay,
Anytime you're
that's good to
given
know.
this codon table, it'll be from the mRNA sequence.
Okay, so whenever it's from the mRNA, it's 5 to 3.
Yes.
And then when we turn that into tRNA, it's 3 to 5 always. Okay.
Correct. Yeah. 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 Every time you're finding that complementary sequence, it's just like the orientation's going to flip.
I was making this so much harder for myself.
No, no, no.
Um,
That's okay. Yeah.
for so for question 13,
Hmm.
what is the N to C? I don't think I understood that part.
So,
What's the
and
N to C or C to N?
gotcha. So that's just how you label like protein sequences. So
Okay.
the reason that like DNA and RNA sequences are labeled 5 prime to 3 prime is because that's like the carbon that like it leads with on like
Oh,
the actual sugar. So
okay.
N and C, I don't know, does that make sense? What I just said?
I guess a little bit.
Gotcha. So when you have like okay, let's see if I can draw while I'm sharing. I can. My mouse is a little weird, so we'll see if this behaves. So you have like a pentose sugar. Ooh, that's not a good drawing whatsoever. But anyways, like you like normally when you're looking at a sugar, you like you can label the carbon. So this is like carbon one, two,
Yeah.
and I'll have like this little apostrophe. So on one end of the DNA sequence, like at the start, this 5 prime is going to be like sticking out from one end
Okay.
because there's no new amino acid. So that's why it's 5 prime. And then like vice versa on the um, end of the actual DNA strand, the 3 prime is going to be the one kind of sticking out. So like it'll be this carbon at the end. Yeah, I encourage you to look that up too because the diagrams that you like find online will be a lot better. But that's just like the orientation anyways, but the N to C
Yeah.
is like they call it like the N terminus and the C terminus because the N
Hmm.
stands for like the amine group that's part of like the polypeptide chain.
Yeah.
and then the C group is part of it's like a carboxyl group that goes on the back end. Let me see. I'm actually going to look up an image. Let me get rid of this. Maybe that would be easier instead of me trying to draw with the mouse. Can you still see my screen, like in this new
Yeah,
tab?
yeah.
Okay, perfect.
Hmm.
See if this comes up with anything good. So just looking at like let's see, an image of a DNA molecule, you'll see that the 5' end is because that 5' carbon is sticking out there, but then the
Yeah.
3' end is going to be this 3'. Oh, sorry, that's not what I wanted. I do like that guy though. And then the 3' carbon is going to be sticking out at the 3' end there. So it's just like
Yeah.
the free group that, or like the free carbon that's there. So then if we look at C terminus, you'll see that that just is part of like the actual polypeptide chain because every like amino acid has like an amine group and then a carboxyl group right afterward.
Yeah.
So that's just what it denotes.
Okay. And so when we have a question like question 13,
Hmm.
like what are we looking for? Like how do I answer this question? I don't understand that.
I see. So when it's asking what are the correct strand ends for the polypeptide chain you built in the above question, that's just
Yeah.
asking what orientation the polypeptide chain is going to be in. So when you're looking 5' to 3' on mRNA,
Yeah.
your polypeptide chain is normally going to follow like a similar orientation. Like you're always, I guess I'm kind of giving away the answer a little bit, but you're always like starting with an amine group on the 5' end.
Right. So because based on the activity one, I understand that when you switch it to tRNA, it switches from 3' to 5'. So I
Yes.
guess that makes sense.
Okay.
So I guess based on this mRNA sequence, we're just following the 5 to 3.
Yes. Correct.
Mm.
Yeah. So then on the polypeptide chain though,
Yeah.
let's see, how do I explain this without like giving away the answer like exactly? You're going to see
Uh,
that like the first amino acid, methionine, the like the if you're thinking like orientationally, like the left side of it is going to be free, and that's where the amine group is. So the polypeptide chain itself is going to follow the same orientation as the mRNA sequence. Does that answer your question at all?
can you just repeat I think the last two sentences that you just said?
Yeah, so the polypeptide chain orientation, like in terms of the N and C terminus, are going to
Yeah.
follow the same orientation as like the mRNA sequence, which is 5' to 3'.
Oh, okay. So, okay.
Yeah.
Maybe now. Okay, maybe I can ask you one question now.
Okay.
So it will still be like N to C then, because it's
I
the
track. Yeah.
polypeptide chain, right?
Yes. Correct. Yeah. Yeah. So
It wouldn't be 5 to 3 anymore. In
correct.
terms of wording, it's going to be N to C.
Correct. Yeah. Yeah. Because you'll use the N to C termini um to like denote the orientation of the polypeptide chain.
Got it. Okay, I
Yeah.
think that, yeah. Okay, that's helpful.
Okay, good. Okay.
Yes. Yeah.
Awesome.
I think that's mostly. So for question 14,
Hmm.
it's saying what elements? So is it like literal elements that's on the table is what we're talking about? Because I know in the actual experiment, we also use the bacteriophages, right? But that's not an element, if
I
that makes sense.
so that would so yeah, it's asking about every part of
Or
it.
every component of the, okay. Okay, when
Yes.
I saw elements, I was like, is it like actual literal elements or just parts of the experiment?
Yes, it'll actually be like all the parts of the experiment.
Okay,
Yeah.
that's
So like,
clearer.
okay,
Okay.
cool. Yeah, I would say like an element is like one of the actual things that they used in the experiment.
Okay. Okay. Okay, that's helpful.
Okay,
That's
good.
clear. Thank you.
Yeah, of course. Would you like to go over any of the other questions or even Isra or Jay? Feel free to chime in too if you guys have specific questions.
I don't really have more so question of the content. I just wanted to know when they meant matches, the term matches. Does that mean complementary or like identical?
Sorry, when what matches?
So there's like a wording on one of the questions that kind of got me confused. One second, let me open
I
Gotcha.
think
it.
it's question 5 you're talking about.
Um, Sorry. So which DNA strand sequence matches the mRNA sequence used in translation
Mm-hmm.
besides the difference? So matches
Oh, I see.
means like identical or complementary.
Matches the mRNA sequence used in translation. That would be like identical.
Okay, okay,
Yeah.
got it,
Yeah. Like if you're
got
looking at the,
it.
yeah, like if you're looking at the DNA strand and
Right,
comparing it to the RNA strand, like they have the same sequence, but the T's are substituted for U's.
use. Okay, got it.
Yeah.
Perfect. Thank you.
Yeah, of course. Good questions, guys. Hey B, how's it going? We're just going over some of the questions. So if you had any questions about the quiz, feel free to ask them. One thing I will mention is that all of these experiments that you guys learn that kind of, I don't know, gave scientists the tools to finding out that DNA is the genetic component that's kind of like transferred from one organism to the next. I would highly recommend that you study those experiments a little in depth and just kind of understand like why they use different elements and how they led to like the conclusions that they made. Because that'll be important for like activities and exams in the future.
Mm. So basically all the experiments, know in detail what happens and why it happens.
Yes, yes.
Okay.
Yeah, especially like yeah, Hershey Chase, the Avery experiments as well.
Yeah,
Yeah.
cool. I think that's all I needed help with. Thank you so much.
Okay, awesome. Yeah, yeah, of course.
Perfect. All right. See you then. Bye.
See you.
For the experiments, is it provided for us on the modules? Like the details is what I'm talking about. Like one
Mhm.
of the questions had like T2 bacteriophages,
Yes,
but none of like the PowerPoint didn't have that or anything. So,
that's a good question. So if the PowerPoint didn't go into it specifically, um, I'm not too familiar with what Dr. Holacek goes over, so maybe Jeanette and Janina might know, but um, feel free to like use Google as well to make sure that
okay.
which type of bacteria was used or bacteriophage.
Hmm.
Um, but the actual, I mean, as you see, like one option, this strep option, is just
Right.
bacteria, and then the T2 is the actual bacteriophage.
Bacteria. Hmm.
Yeah, yeah. So that's like, I guess you don't
I
really
thought
need.
it was a trick question for a second because it didn't like none of the ratings, or she didn't even go over like what type of bacteriophage
Mhm.
she was talking, like that was explained during the lecture, the video lecture. So I was kind of like, oh, is this a trick question?
Gotcha, definitely.
Yeah, yeah, yeah.
Yeah, I think this question just includes those details just to give you some extra information about them. But yeah, I wouldn't worry too much about the actual name.
Okay.
Yeah, bacteria or bacteriophage used. Yeah.
Okay, thank you.
Yeah, of course. Yeah, of course.
Hi,
Yeah.
I have a quick question.
Yeah, what's up?
Can you please explain question 13 for me? I'm kind of having a hard time understanding what goes at the end.
Gotcha. So looking at the like the ends of a polypeptide chain, um, you'll see that like it starts, or usually like the polypeptide chain, looking at it from like a 5' to 3' orientation going left to right, it's going to be like the N to C orientation. So another student asked this question too, so I brought up this picture, and you'll just see that like if you're looking at a polypeptide chain that just has like three amino acids, um, the one on the left is going to be the N terminal side, and then the other one's going to be the C terminal, and that's just because the amine group is like the end over here, and then this one has the carboxyl group. I don't know if that was your question.
Yes, that did. Thank you.
Okay, yeah, yeah, of course. Yeah, anytime you're looking at a protein or polypeptide chain, you'll be using N and C terminus. So when you're working on activity one, instead of labeling it, 5' to 3' or 3' to 5', however, um, you write your answer, it'll be N to C or C to N instead.
Thank you.
Yeah, of course.
If possible, could you also explain question 3 a little more?
Oh yeah, for sure. This was also another question that students were asking about. So, which DNA strand sequence match the mRNA sequence used in translation, besides the difference between the thymine and uracil bases? So, this question is just asking when you're looking at the DNA strand and like the two different DNA strands and comparing them to the actual mRNA sequence, um, which one or like what terms here are um, like kind of explain or are different names for the strand that kind of runs the same orientation as the mRNA strand. So that'll be the strand that kind of goes like 5' to 3', since that's what the mRNA goes as well. So you'll notice that there's going to be like two separate names for that DNA strand that goes 5' to 3'.
Thank you.
Yeah, of course. Did any of you have any questions about any of the information that was brought up in the PowerPoint video?
I have a question about the amino acids and the codons.
Hmm.
So if there's like 15 amino acids, are they still going to be encoded by 61 codons, or does that like divide by a certain number?
I see. So you're saying if there's 15 amino acids, um, actually, what were you saying about the 61 codons? Or were you like thinking nucleotides?
Yeah, yeah. Thank you.
No, no, you're good. Yeah, so just whenever you have a question like that, you'll be used looking at like the number of nucleotides and then dividing that by three to get the number of um, amino acids. I don't know if that was your question.
I think so.
Okay. Yeah, because I don't know if you're talking about which question was it? Like a question like number two, where it says like how many amino acids would be coded from a sequence of 60 nucleotides.
Oh, yes, I forgot that was even a question.
You're good. So the important thing to keep in mind here is that, so I mean, you would divide by three, like the number of nucleotides, you would divide that by three, but that last codon isn't going to encode for an amino acid. It's just going to be a stop codon. So I guess you would do like, you would take off three nucleotides and then divide that number by three.
So you would just like take that number you're given minus 3 and then you would divide it.
Yes, correct.
Okay. Okay. Thank you.
Yeah, yeah, of course. Yeah, make sure in activity one too that you notice like if there's a stop codon or whatever that you just write stop there for that, and that'll be in different activities too. But yeah, that stop codon never encodes for an amino acid, so it won't be like actually part of that polypeptide chain that comes out of it. So good question. Yes, B. I will still be on this same Zoom link tomorrow from 9 to 10 to go over activity one. And then if you can't make that one, I think Aaron has office hours from 1 to 2 p.m. AZ time as well. So yeah, feel free to join those and we'll answer your questions too. And then we always make sure to record our office hours so that if you can't make them, because all of you usually have stuff going on outside of this, We usually upload it somewhere so that it's accessible for you guys and you can reference them later on as well. Thanks, U2B. Yeah, of course, Kim. Have a good one.
Um, uh, okay. So I'm doing the recitation or whatever you call it.
Hmm.
And I had a question real quick on a few of them, but the big one would be on 13.
Yeah, that sounds good. Yeah, we can go in order too if you want to, if you had questions before that one.
Well, that's like the big one, I guess, right now.
That sounds good.
I just didn't see that, like, I don't know if I'm just not looking at the right spot, but I didn't see that in the video or in the little one where you do the quiz or whatever for where she goes over it. I didn't see anything about the polypeptide chains ends and what they're called. Like,
Gotcha. Yeah, no, that's okay. There were just like 8 students here and they were all asking about it also. So maybe there is a gap somewhere, but what I was showing them was this image on Google. So here, maybe let's look at this one actually. That's kind of cool. So looking at like a polypeptide chain, kind of similar to DNA where you have 5' and 3' ends just based off of whatever carbon on the ribose sugar is sticking out. You're going to have N and C termini because of the chemical group that's on the ends of the polypeptide chain. So looking starting on the left side over here, you're going to have the N terminus with the amine group. Now on the right side, you're going to have the C terminus. And
okay.
yeah. So when proteins are translated from mRNA sequences, you're always going to start with the amine group, like that methionine, like the ATG
Hmm.
codon for methionine. It's always going to have its amine group on the left side.
Okay.
Yeah.
Alrighty. Well, I think that's really the only real question I had, I think. Let me just scroll through real quick. Oh, for question 14,
Hmm.
so it talks about the T2 bacteriophages.
Yeah.
Um, in both, I believe they only mentioned there being bacteriophages.
Right.
Um, is that like more specifically they were T2 ones and they just didn't mention that, or is that supposed to be something completely different and I shouldn't click that?
No, you're good. It's just a more specific term for the type of bacteriophage they use. Um, since us grad TAs kind of put together the recitations, we nerd out and like get specific with the name. So yeah, you don't have to worry about the name itself or like get thrown off by
So so that just indicates general bacteriophages, and I don't want to be like, would that be right? But
Yeah, well, yeah, you're on the right track, I'd say.
okay, cool. I think everything other than that, I mean, it was pretty clear in the videos and in the general note things. Is there like a textbook link that I'm just missing that I should be going over this stuff into, or should I really just be mostly going through those?
I would just mostly go through those. Jeanette and Jonina, since you guys have taken the course before, did you guys ever use a textbook?
So back when I took it, it used to be called books, but now everything is in the Canvas module. So all the readings, all the lectures, all the concept quizzes, those are in your modules. So just make sure that you're going through all that. And that's basically what everything's going to be tested on is in those modules.
So really just focus in on those and don't, you know, look for additional stuff.
No, I mean, you can if there's like a particular topic that you're really struggling with, it can be helpful to either discuss it like in the Discord or, Google it. As weird as it sounds, that can kind of like help you get a better handle of it. But everything that you actually need to know is somewhere on within the material that's on Canvas. You don't need anything additional.
Uh-huh. Well, actually, to be honest, I did Google 13, that first one I had a question about because I didn't see anything about it. And then I was like, I started overthinking and stressing, thinking maybe it's like a test or something to expose who's Googling. And,
No.
if I get it right, I'm going to be in big trouble. So I figured I'd ask since, it's available right now and I happen to be here right now. So,
Yeah, yeah. I mean, the office hours are generally a better source of like good information, but if it's like 2 in the morning and you can't like ask anybody, I would go on Google or GPT or something and kind of just discuss it to try to figure it out.
alrighty. Well, I think that's all I got for questions. If I find anything else, I'll just show back up over here. But
Perfect. Yeah, yeah. I'll be hanging out on here for another like 25 minutes. So
alrighty, well, enjoy your 25.
thanks. Thank you, Ryan. Have a good one.
All right, you too.
Thanks.
Are they watching the rec video?
It kind of doesn't sound like it.
I'm sitting here.